Welcome back, and welcome to part 3 of the slideshow. I thought in this last little bit of the slideshow, which is not as long or as extensive as the other two, I thought I would just introduce you to some of the types of microscopes that we use to look at microbes. You are familiar with some of this from first year and from other courses you may have taken. So for those of you who are familiar with this, this will be a bit of a review, but let's just take a look. Okay, so we're going to learn the difference between light microscopes, fluorescent microscopes, and electron microscopes. All right, so light microscopes are sometimes referred to as bright field microscopes, and they're called bright field microscopes because they kind of basically, when you look at the slide, you put a slide in between your eye, or you put a slide in between the objective lens and the light source, and you're essentially looking down into the light bulb of the microscope, and so the field that you're looking at your specimen in is bright. So it's called a bright field microscope. And these images are easy to recognize because the background is white. Right? You're looking at a bright field microscope. The common name for it is a light microscope, and the background, the field, is white. All right, a fluorescent microscope uses, instead of using visible light as a light source, a fluorescent microscope uses ultraviolet light as a light source. The human eye cannot see ultraviolet light, and so the background appears to be dark. But the specimen appears to be fluorescent, which it is, because you've stained it with a fluorescent dye. All right, so that is an example of fluorescence microscopy. And there are various ways that you can get the dye to stick to the specimen that you're looking at, but this is a characteristic of fluorescent microscopy. Fluorescent microscope images are easy to spot. They're easy to recognize because the background is dark and the actual field, the subject, the specimen that you're looking at, appears to be fluorescent. Okay, then we have a powerful type of microscope called an electron microscope. Electron microscope, instead of using visible light or instead of using ultraviolet light, it uses a beam of electrons to illuminate the specimen. Generally, the resolution limit is defined as the smallest degree of detail that you can see. The resolution limit is proportional to the wavelength of the light that you're using to look at that specimen. Right, so visible light is in the visible light wavelengths are in the order of 600, 500 nanometers. So that's like 0.5 microns. You know, remember that there are 1,000 nanometers in a micrometer. So if visible light is, say, 400 or 500 nanometers in wavelength, that means that the smallest detail that you can see will be about 500 nanometers. 500 nanometers is half a micron. And so therefore it stands to reason that when you're using a light microscope, the smallest thing that you can see is around, the smallest detail that you can see is things that are around 0.5 microns, which is about the size of a bacteria. Fluorescent light, you have a little bit smaller detail down to around 200 nanometers for ultraviolet light. And so you can see things that are around 0.2 nanometers with a fluorescent microscope. So you see a little bit more detail with a fluorescent microscope than you do with a regular microscope. But an electron microscope has light wavelengths of around 1 nanometer or less. And so what that means is that with the electron microscope, you can see details down to 1 nanometer or so. So when we want to look at really small things, when we want to look at the interior of a cell, we want to look at a ribosome, or we want to look at the Golgi complex or anything like that, we tend to use electron microscopes. There are two types of electron microscopes. One is called a scanning electron microscope, or an SEM. A scanning microscope looks at the surface of a very small thing. And a transmission electron microscope is where you cut things up and you look at a slice through the thing. So whatever you're looking at, when you see the surface, it's a scanning electron microscope, SEM. When you see a slice through the middle of it, it's a transmission electron microscope, or a TEM. And Generally, those types of microscopes are quite expensive, but that's what they use. And also, those types of images, the images generated by an electron microscope, are always in black and white. So you notice that the two images down here at the bottom are in black and white. Okay, light microscopes, they look like this. The one on the left is one that we would typically use in the lab. It costs about $1,000 each, or $1,200 each. But you don't need to be that expensive. The types that children play with, you know, I had this one on the right. I had one of those given to me by my parents when I was 7. Instead of using a light source, instead of using a light bulb for a light source and electric light, these cheaper versions simply use a mirror to focus sunlight on the specimen. But in both cases, you have a set of ocular lenses. You have an ocular lens where your eye goes. You have an objective lens that you position over the slide, and then you have a light source down at the bottom, and then you have a stage, which is this flat piece, and you put the slide onto the stage, and then the light goes through the slide into the objective lens and then into the ocular lens, and you get magnification. Okay, it's easy to recognize light microscope images or bright field microscope images because they have a white background. That's essentially because you're looking at the light bulb that's below the specimen. And in fact, if you slice things up and you look at, if you slice up cells or tissues or something like that and you look at them, you won't actually see anything because they're so thin that you can't see anything. So whenever you're looking at things with the light microscope or the bright field microscope, what you have to do is stain those specimens. You have to take some kind of a chemical stain that stains the specimen. Now, we've been using bright field microscopy, we've been using light microscopy for about 500 years now. And so we've gotten very good at making the chemical stains that are used to stain the specimen. The very first stains that were used 500 years ago were vegetable stains, where you take, you know, colored vegetable oils and colored vegetable juices, and then you stain the tissues and then you look at them. And, you know, in the last 200 years or so, we've become very sophisticated at making chemical stains. And the chemical stains are usually, just for reasons I won't go into, the chemical stains usually stain things either red or blue. And if you mix together red and blue, you get kind of a brown color. So looking at a bright field microscope image is kind of, looking at a bright field microscope image is a little bit like looking at a child's finger painting. Right? You know that children like to spray or squirt blue paint and red paint onto a piece of paper and then smear it around with their fingers. And if you smear it around, you end up, if you mix around blue and red, you end up getting purple, and then if you add a couple other colors, you get kind of a brown color. So bright field microscope images are easy to recognize because they are on a white background, and they have either the stained specimens are either blue or pink or brownish in color. Okay, a fluorescent microscope is the same as a bright field microscope, as a light microscope, except that instead of using visible light, it uses ultraviolet light. This is a fluorescent microscope. It costs about $10,000, so it's about 10 times more expensive than a regular microscope. And so here, the construction is essentially the same. You have an ocular lens right here, and you have this is actually a camera up here so you can take pictures. And then you have your objective lenses down here, and you have the slide down here, and then you have a regular light bulb here. And in this box that's behind the microscope is an ultraviolet light. And so the ultraviolet light goes down this tube, and there's a prism that directs the ultraviolet light down onto the specimen. And of course, you have stained the specimen with fluorescent dyes. Now, what fluorescent dyes do is they absorb ultraviolet light. Ultraviolet light is transmitted at a short wavelength, which is too short for our eyes to see. Fluorescent chemicals absorb that light and then re-emit some of that light at a longer wavelength, which we can see. Right? So a fluorescent dye is something that absorbs ultraviolet light that we cannot see and then re-emits it at a longer wavelength in the visible spectrum so that we can see it. So the fact that we have the specimen is being illuminated by ultraviolet light means we can't see the ultraviolet light, so the background appears to be black. And the specimens that we're looking at have been stained with fluorescent dyes. Fluorescent dyes usually stain things either yellow or red, or sorry, green or red. For example, fluorescein is a dye that's very commonly used for fluorescent microscopy, and it stains things green in color. And then there's another dye called Texas red, which, as you would guess, is red in color. It fluoresces red in color. So you have a dark background and a fluorescent specimen, which always kind of looks a little bit like a fluorescent advertisement sign, like a big Coca-Cola sign or something. So fluorescent images are quite easy to look at, are quite easy to recognize as well. All right, an electron microscope uses a beam of electrons to illuminate the specimen instead of a beam of light or even ultraviolet light. This is an electron microscope. It costs typically $100,000 or $200,000. So these are about 1,000 times more expensive than, or sorry, about 100 times more expensive than the microscopes that we have in the lab that cost $1,000 each. So this is worth the cost of 100 microscopes. Typically colleges don't have them because they're too expensive, but research institutions do. Universities where scientists are doing research will take the money for an electron microscope, but usually because they're so expensive, individual laboratories do not own their own electron microscope. Usually a university department owns the microscope, and then individual professors within that department will pay to use it for a certain amount of time every day. Right? So the department or the university owns the microscopes, and then the professors have grant money from to do research, and they pay some of that grant money to the university in order to let them use the microscope. And usually you have to be specially trained to use it because it involves high voltage and a lot of complicated equipment. So this is what's known as an electron tube, and at the top of the tube there's an electron gun that fires a beam of electrons down this tube, and the electron beams are spread apart by copper magnets instead of by glass, and so that's how you magnify the image. You spread the beam of electrons out using magnets instead of lenses, glass lenses, and the image always comes out black and white on this computer screen here. Right, so the images from electron microscope images are always black and white. Okay, so this is an example of scanning electron microscopy. What you're doing here is you're deliberately looking at the surface of whatever you're looking at. This is a flea, so it's quite small, but it's not terribly small. You know, a flea is a multicellular organism that's made up of lots of cells, and so we're not looking at individual cells. But it is true that you can use a scanning electron microscope to look at cells. You can use a scanning electron microscope to look inside cells as well. You just have to break them open first. Now, it looks kind of like this flea has been spray painted with silver spray paint or something like that. And in a way it has, because in order to see things with the scanning electron microscope, you have to spray them with vaporized metal. And so the electron beam will actually pass right through the surface of a flea or a cell without stopping. And so if you want to see it, you actually have to coat the surface with metal. And so you use a machine called a sputter deposition machine that vaporizes gold or silver and sprays a coating on the surface of whatever it is you're trying to look at. So there's a common misconception that when you use scanning electron microscopy, you're looking at big things like fleas, but in fact you can use the same principle to look at small things like cells. Okay, so the images for scanning electron microscopy are always black and white. They look kind of like something has been sprayed with silver spray paint. The specimen has actually been electroplated with metal, and the image appears to be 3D. The scale of these images is typically very small, anything from 1 nanometer to as big as you want. Obviously a flea is about 1 millimeter in length, so that's huge by microscopy standards, but you can look at anything from 1 millimeter upwards. Alright, this is a transmission electron microscope. This is where you've taken some tissues and some cells and you've cut them up into thin slices, and then you stain them with a metal. In this case the metal is called osmium tetroxide. And so you stain them with a metal salt, and then the electron beam passes through the specimen, if you didn't stain the cell with a metal salt, the electron beam would just pass through it and not see anything. So we stain the structures inside the cell with osmium tetroxide. So here you can see the nucleus of this cell. There's the nucleus, right? And here you can see the rough endoplasmic reticulum and the Golgi complex over here. So this is an example of transmission electron microscopy. Again, transmission electron microscopy, or TEM, uses the images that are generated appear to be black and white. It appears to be on a light background because you actually are looking through the cell again. The specimen is sliced and stained with osmium tetroxide. Again, the scale is very small. You can look at anything from a small down to a scale of 1 nanometer all the way up to 50 microns or something like that, or even a millimeter if you want. Finally, we said that the electron microscope images are always in black and white, and that's true. But if sometimes a researcher will deliberately color the image to make it easier for the viewer to see different structures, and so that's what's happened here. Here we have the original image. Do you remember what this is? This is the influenza virus. Somebody has taken a special type of transmission electron microscope image, and they have then colorized it with Adobe Photoshop, but you could just as easily use crayons or colored pencils or water paints if you wanted. And so you've colorized the black and white image, right? And that is referred to as a false color electron microscope image, a false color TEM. Everyone knows that these images are taken in black and white, so it's not like they're cheating or trying to deceive you. Everyone knows that they have colorized it, but they just colorized it to make it easier for you to see different parts of it. Okay, let's see if you can remember or if you can tell me which type of microscope took each of these images, just so you can recognize microscope images. So what I want you to do, I'll show you an image, and first you tell me what type of microscope image it is. That's the easy part. And then tell me which type of organism we're looking at. Is it a bacteria? Is it a protist? Is it a yeast? Is it a helminth? Right? Is it a fungus? And then the hardest part, possibly, tell me if you can name it, because these images you've seen already in part 2 of the slideshow. So let's just see if you can do those exercises. So first of all, what kind of an image is this? What type of microscope took it? The answer is that this is a bright field microscope image. It's a light microscope. Okay, can you tell me what type of an organism this is? If you said helminth, you would be correct. This is a worm. It's a nematode worm. In fact, do you remember specifically which nematode this is? I wouldn't ask you to identify things by sight on the test, but just for fun, can you tell me which organism this is? Do you remember? This is Trichinella spiralis. Trichinella spiralis. All right, what type of microscope took this image? These images were actually taken by a scanning electron microscope. It's a little bit hard to recognize because they used Adobe Photoshop to cut the image away from their background and then pasted them together on a black background like this. So in real life, if they hadn't manipulated the image, all of these little things would be on a background. And so the background would make it easier to tell that this was actually a scanning electron microscope image. So these are 6 independent SEM images that they've simply cut out and cut and pasted onto a black background. Okay, do you remember what these organisms are? In fact, these organisms are called dinoflagellates, and we'll talk about dinoflagellates. These are the creatures that create red tide poisoning and so on. They are a type of protist, by the way, protists. Okay, what type of microscope took this image? Right, the answer is, because this thing looks like it's fluorescent, this is a fluorescent microscope image. Can you tell me what type of an organism it is? I'll tell you that these are nuclei, right? And each of these cells contains 2 nuclei. So what are the only organisms that you know of that have, sometimes, depending on the species and the genus, sometimes they have more than 1 nucleus? These are protozoa. These are protists. Can you remember which one specifically? These are actually Toxoplasma gondii. Toxoplasma gondii. Okay, what type of an image took this? It's not much of a challenge because it's actually written on the image. You see down here, LM stands for light microscope. You can tell the scale. So these cells are approximately 10 microns. These are, and then you can see that they have 2 nuclei. Right? So these are Giardia lamblia. All right, what type of an image is this? What type of microscope took this image? Okay, if you guessed light microscope, you're correct. Right? And what type of an organism is this? All right, if you guessed that this is a helmet, again, you would be right. And which one is this? This is Diphyllobothrium latum, the tapeworm that will infect fish. It is carried by contaminated fish. So this is a light microscope image. What about these? Okay, if you guessed that this is, again, scanning electron microscope images that were simply cut out and then pasted onto a black background, that's correct. This one down here at the bottom middle has not been cut out and pasted directly. You can still see the background. And these are diatoms. So this is a type of protist. This is a type of protist that lives in the ocean and has a silicon body. What are these? And what type of microscope took this image? Okay, believe it or not, this is a false color SEM. Right, so here somebody has taken the background and used Adobe Photoshop to select the background and then make the background dark, and then they colored these cells in the foreground in kind of this orangey color, this yellow, orange, beige color. Do you remember what these are? These are yeast. Specifically, these are budding yeast. Right, so you see that these cells are in the process of dividing, and there's a small piece, a bud, and there's coming off of a larger cell. So what is the only budding organism that we learned already? Right, so if you guessed Saccharomyces cerevisiae, you would be right. So this is a false color SEM. What about this? If you guessed that this is also a false color SEM, you would be correct. All right, so somebody took the background and they stained it purple, and they took the foreground and they stained it, or they took the cells and they colored them blue with Adobe Photoshop or some other computer program. Okay, now what about the morphology of the cells? What shape are they? Okay, if you answered bacillus, you'd be right. And so what was the number one bacillus that we learned about? Okay, Lactobacillus acidophilus, that's what this is. What type of microscope? Obviously, this is a bright field microscope image, and these cells, what about their morphology? How would you define the shape of these cells? Okay, these are cocci, and what about the arrangement of these cocci? Okay, they are clustered together, so what do you call cocci that are clustered together? You call them staphylococci. So this is Staphylococcus aureus, and this is a bright field microscope image, a light microscope image. What are these? What do you think? What type of image is this? Okay, by now you should be getting used to these. This is a false color SEM image. No microscope can take a picture like this without it being colorized. And these are cells that you would find on the skin. These are in fact Candida albicans, a yeast. What about this image? What type of microscope? Okay, this is a little tricky and a little bit devious on my part. This image is in black and white, so you might guess that it's transmission electron microscope, but in fact it's in black and white just because the photograph is in black and white. So this is a black and white photograph of a color image, and it's actually it's a bright field image. It's a light microscope image. Okay, now these are bacteria, and notice that this clear zone here is an endospore. These clear zones where you have endospores, the endospores are located on one end of the bacteria. What about the morphology of the bacteria? If you said it was a bacillus, you'd be correct. And we have a bacillus that is making endospores, and those endospores are located at one side, at one end of the bacteria. There are other bacteria that make other bacilli bacteria that make an endospore that's located in the middle, and that's one way that you can tell one species from another. is by the positioning of the endospore. But what were the bacteria that I told you about that make endospores? The main ones, or the first ones that we learned about, were Clostridium botulinum, and that's what these are. So this is a bright field image of Clostridium botulinum, which is a bacillus. What about this? Yes, this is a transmission electron microscope image, and again, this is our old nemesis, the influenza virus. What about these? Okay, if you guess this is a bright field image or a light microscope image, that's true. What kind of cells are these? What shape are these? These are bacteria. What shape are the bacteria? They're cocci, right? And how are they arranged? They are arranged into chains, right? So therefore, what would you call them? You would call them streptococci. And what are the only streptococci species that you learned of? Streptococcus pyogenes, and that's what this is. It's a light microscope image of Streptococcus pyogenes. What about this? This is a false color SEM. It happens to be a false color SEM of Candida albicans. And what's this? This is a false color image showing our old friend, a false color SEM. It even says SEM down there. And you can see the scale. So this is a fairly large cell. And this is our friend a macrophage, which is a white blood cell whose job it is to eat these little bacteria, these little bacilli in this case, and form pus in the process. All right, so that's enough for one lecture series. Anytime you want, you can proceed to lecture 2, which is a more thorough examination of bacterial morphology. So we're going to be discussing bacteria next, and how you determine which bacteria is which based on the shape, what types of words we use to describe different shapes of bacteria and different arrangements of flagella, and so on. Thank you.